lung tumours. There are a number of reasons why in the B16 model this might be so. There might be immunological responses against this altered cell, even although it doesn't have an altered protein. And there is some evidence that Rho0 cells have increased class 1, which might stimulate immunological responses, and then receptor 1, it can be increased as well. So we decided to repeat these experiments in immunocompromised non-skid mice, which are often used in xenotransplantation studies. So again, the wild type and the mitochondrial gene knockout cells both grew tumours. When they got established, the rates were about half. The delay was a little longer, about 30 days with the Rho0 cells, but they still formed tumours. Looking at the intravenous injection, these cells again did not form lung tumours. Out of day 64, there were no lung nodules compared with the wild type. So the question arises then as to why these B16 melanoma cells without mitochondrial DNA and are purely glycolytic fail to form lung tumours. And one of the reasons is that they clearly will have low ROS production. They have no ability to produce reactive oxygen species in the mitochondria. And that signalling may in fact be required for some of the early events in metastasis. And one of the early events is when cells break away, when epithelial cells break away from their local environment. Normal cells will undergo a programmed cell death. Tumor cells have adapted and will not undergo programmed cell death. So what has happened in the Rho0 cells in this state? I guess altered invasion. There are other explanations as to why these cells might not form tumours in the lung, but we decided to start off at a likely early point in the signalling cascade. And some of this has not come out, but it just shows that tumour cells have a ROS-dependent reversal of what is called anoikis, or adherence or attachment dependent programmed cell death. I guess that's the second part that does show it all. So we looked at these cells in a couple of culture systems. One is a two-layer agar system. Parental cells under these conditions will form spheres of cells very much like solid tumours. The Rho0 cells fail to form colonies under these situations. Another assay involves a stem cell, cancer stem cell-like assay, where the cells are grown in serum-free medium with fibroblast growth factor and EGF. And under these conditions, the parental cells form what are called spheres, small spheres, but they are certainly consistent and high frequency. The Rho0 cells fail to form these. However, under conditions of fetal calf serum, both cells grew quite well, although the Rho0s were a little slower, as I mentioned. So one of the arguments then is that reactive oxygen species themselves may be promoting this resistance to anoikis that we see in these Rho0 cells. And this has been looked at in other cells, and ROS has been associated with this process of detachment-induced apoptosis or reversal of this. So we looked at the intracellular ROS production in these Rho0 cells using a mitoSOX marker. The Rho0 cells, as we would have expected, produced very low amounts of reactive oxygen species. And this is not only in the Rho0, B16 Rho0, we have in our lab 6 other human and mouse mitochondrial gene knockout models and other models which have very low reactive oxygen species. Looking at the peroxide production or intracellular peroxide, again, this was down in each of the Rho0 cells we looked at. You heard a little bit about N-acetylcysteine as an antioxidant yesterday. If this is added to the wild type cells, it actually reverses the ability of these cells to form clusters and the cells curl up and die. So it appears that the active oxygen species are an important component of the ability of these cells to spread and grow, to invade the local tissue. So in summary, the mitochondrial gene knockout model that I've described is associated with delayed tumour growth in 2 different mouse models, one of them being immunocompromised. These cells fail to form tumours in both wild in lung tumours when injected intravenously in both wild type and immunocompromised mice. Failure to form these tumours may be due to this anoikis or the detachment-mediated programmed cell death that I mentioned. The ROS plays a key role in transducing pro-survival signals. It is an important signalling molecule, and it appears to be associated with progression and metastasis, at least in some models. So in conclusion then, the glycolytic tumour model cells have failed to metastasize, and this may be due to events in the early metastatic process involved with signalling due to reactive oxygen species and peroxide. And I guess we're now setting up to look at whether this is a common phenomenon in other tumour models. In breast cancer, we're beginning to set up the 41 model and we'll investigate whether or not the mitochondrial genome has similar, knockout has similar characteristics in this model. Thank you.
About mitochondria in these cells. Do they form mitochondria on the nuclei?
Sorry. Yeah, I should have gone into that a little bit. There are 13 genes in the mitochondria that encode essential components of complex 1, 3, 4, and 5 of the